Hi everyone, my name is Nico.G. Galasso. I'm in the MCB 540 class. I'll be just talking today about anthocyanin biosynthesis, DNA methylation dynamics in sweet orange fruit. Let's get started. Overall, I'll be talking about six main points. I'll be introducing what anthocyanin is. I'll be giving a background of the evolution of citruses. I'll be going over RT-PCRs, RT-PCR, and MSAP, all used in a study I discussed in this video. I'll also be going over blast and analysis, DNA methylation dynamics, and of course conclude and go over the scientific merits of the studies. So let's talk with the introduction. So what is anthocyanin? Anthocyanin is a pigment found in a variety of fruit and vegetables. It is found in the color variation seen in multiple different plants, radishes, red potatoes, cabbages, purple sweet potatoes, black carrots, all things up to hibiscus sativa and blood oranges. Anthocyanins act as a stress mitigator and they aid plant growth and development by increasing anthocyanin accumulation, which leads to increased resistance to various biotic and abiotic stresses. Some of those abiotic and biotic stressors could be reactive oxygen in the plant cells, can also act as an absorption for excess light, which is why you see the peels be darker skinned. They also play a role in signaling pathways involved in stress responses, regulating gene expression and during stressful situations for the plant. Overall, anthocyanins contribute to plant resilience by enhancing tolerances to various environmental stressors, including some as drought, UV radiation, and even pathogen attacks. Let's go over to the next. Let's talk about a little bit about the background of the evolution of citrus. If you're not currently aware, now you know, but in a study in 2018, Wu and Null actually did a genetic work on the phenology group of all citrus sinensis groups and found that the sweet orange, what we normally call an orange at a supermarket, is actually a hybridization between a female pomelo and a male mandarin. And the study described that all citrus diversity within the sweet orange, sour oranges, and even grapefruits can be traced back to our hybridization and mutations from two ancestor citrus plants, mandarin and the pomelo. Genetic structures and heterozygosity of the citrus species see different types of sweet oranges, limes, citrus to come from this hybridization. And in a study by Cecilia et al. In 2020, they actually did a principled coordinate analysis of 58 different citrus ascensions based on the pairwise nuclear genome distance and metric multi-scaling. The first two axes actually show a separate of the three main citrus groups, citrons, pomels, and mandarins, with the interspecific hybrids of oranges, grapefruits, lemons, and limes situated at intermediate positions relative to the parental genotypes. Next, we're going to talk a little bit about the different varieties of sweet orange, specifically the ripening periods. You might not be aware, but there are about 16 and more different varieties of sweet oranges. You might be known to the navel that you might find on the supermarket all the time, or the Leah, but there are also very different types. There are different types of sweet oranges and ripening periods. Sicilia and all actually use a specific type of Tarocco tapi, using QRT-PCRs and RT-PCRs to indicate what genes are expressed and to quantify those gene expressions. Tarocco tapi actually looks very much like most other Tarocco sweet oranges, where it has a deep reddish hue, and this is actually from the anthocyanins. So let's talk about the actual tools that they were used during these studies. So the first one was RT-PCRs. The RT indicated whether particular genes were actually expressed or not in pulled flesh from the 60 different Tarocco tapi fruit during a cold study. They found that PAL, CHS, DFR, ANS, and UFGT were all found to be highly expressed in the high pigmented genes versus the no pigmented genes. There was three different tests of 15 days of cold storage, 10 days of cold storage, and 10 days of cold storage, and finding 3 to 4 times higher expressions of the genes mentioned than in non-pigmented. They also did the RT-PCR to observe which genes were expressed, and you can see here an actual overall graph showing the elevated high pigmentation in A, B, C, D, and E, which again shows each of the different 5 genes I mentioned earlier. The team also did a QRT-PCR. The QRT-PCR was used to collect gene expression levels for genes that play a role in a biosynthesis pathway, specifically for anthocyanins that were of interest. They found that those genes of interest were again found to be the same that were highly elevated in a high pigmented fruit versus non-pigmented fruit. Again, PAL, CHS, TFR, ANS, and UFGT were all tested again in the 60 Toro-Kotapi oranges. Lastly, the team wanted to do a methylation sensitive application polymorphism, which is a long way of saying they wanted to test for and detect methylation in either promoter regions, gene bodies, and genetic regions, and repetitive sequences throughout the genome. As seen on the left, you can see the following structure of genomes to selected PCR applications, all the way to fine DNA data analysis. And Cecilia and all uses MSAP to detect methylated regions in high pigmented and no pigmented flesh, discovering once again our 5 friends, PAL, CHS, BFR, ANS, and UFGT. You must now be wondering about the insight to this methylation status that these genes provide. They provide valuable information about their regulation expression in response to environmental factors. Seeing that cold stress 10, 15 days and more led to higher amounts of these genes being expressed in the actual during those PCR tests, we now can think about how these patterns could be associated with key genes involved for anthocyanin biosynthesis. So I specifically did a blast comparison to other citruses besides just Toro-Kotapi, which is what the group did with the PCR test. They were looking very much in what they found from the specific oranges they used to test. And I thought, what about all the other citrusinensis varieties that exist in the world? There are so many. So taking those different genome sequences that they found, I used BLAST to determine if those matches and the other varieties, knowing that all citrusinensis come from an unknown ancestor in Asia. So during the BLAST, I found the following. There was carrier coverage of 100%, 94, e-values of 1.3 for CHS. This tells me possibly that it is relatively low, suggesting that match is likely, and not due to random chance. All the other sequences found significant e-values and high coverage of 100% or more for DFR, ANS, UFGT, and Ruby, and DME, and lastly DMO1. All these, and lastly Ruby Pro2, can't get that one. All these different genes were found to have different functions going through NCBI's processes to do gene analysis. You can go through and click on genes and it'll tell you what it'll do. During that, I found that each of these different 5 genes that I have on the screen were directly involved in the regulation of anthocyanins and, of course, flavonoids. So the next question is, well, how did these anthocyanin regulatory genes get turned off if most of them are methylated, as seen in the previous section? Well, let's go over what DNA methylation is. So if you don't already are aware, DNA methylation dynamics refers to the change in patterns of DNA through the closing of compactness of chromatin compaction for transcription silencing. It allows the genes to be read by their promoter regions and transcribed, or it causes them to be closed. And cysteine methylation is one of the most common versions, or CpG denucleotides regulate this expression by influencing chromatin structures. And to reiterate, these epigenetic modifications, like DNA methylation, are reversible, which we see in the process of Demeter-like-1 or DMO1. This is one of the genes I found on NCBI that when going through and clicking it, you actually can go through the genes and see what it does. And sure enough, gene ontology tells us that it enables DNA demethylation activity, involved in specifically DNA demethylation. This is really interesting, and Cecilia and all noted that a significant reduction in cytosine methylation levels in high pigmented fruit segments exposed to cold stress, where they found higher levels of the methylated groups in no pigmentations, with DML1 along with all the other demethylation enzymes also being lower in those groups. So, last quick little rundown, look over the CPG's islands. This is a quick little glance at all the different genes, and I just highlighted in bold all the ones in cytosines, and it's really interesting to see that all these different rows had been picked up by MSAT, again, methylated groups. Very cool. So part 6, let's talk about the conclusion and the rundown. In conclusion, the investigation into anthocyanin biosynthesis and DNA methylation dynamics in sweet orange fruit under cold stress conditions shed light on the intricate mechanisms governing plant response to environmental stimuli. The study finding underscore the crucial role of gene expression regulation and epigenetic modification in orchestrating fruit pigmentation and stress adaptation. By including the molecular pathways involved, this research not only deepens our understanding of plant biology, but also offers practical indications for agriculture. I'll go over what that means in the next slide. So, critical evaluation. First, the study demonstrated strengths in its comprehensive experimental design. They went through RT-PCRs, DNA methylation analysis to see which the genes were done, using mostly molecular gene processing. However, the potential weakness includes the limited factor of the sample size, only 60 Toropo-Tappy oranges, which may impact the robustness of the findings. They also didn't create a genetic library to really test against all other genes in the citrus sinensis groups, which I found through NCBI and some other websites. But the broader significance understanding the genetic mechanisms underlying anthocyanin biosynthesis in citrus fruits contributes significantly to the field of functional genomics and plant molecular biology. Super interesting. And one of the things I would really want to talk about is that while the conclusions drawn from the data are supported, further validation through functional studies could enhance their reliability, such as using CHLP sequencing could be used to identify DNA binding proteins associated with regulatory regions of targeted genes to really see what's going on between interactions of these demethylation groups and the processing that allows for anthocyanin pathways. Moreover, they have indications for targeted breeding programs aid at improving fruit quality, such as color, antioxidant content, thus offering potential benefits for human health and nutrition. And a recent study, actually by Kuranal, they looked into how the plant tolerance stressors in the native habitats, such as drought, low temperatures, pathogen attack, high temperatures, and even salinity due to looming threat of global warming, could pose a threat to plant growth and productivity, not just for our agricultural sectors, but just for like feeding the nation's worlds and even small areas. A really insight into regulatory pathways also involved in fruit pigmentation and their interactions with environments could, just like cold stress, see implications in the future. Here are a list of my references. I really hope you enjoyed your time viewing this, and I look forward to looking into the discussion board posts, questions, and all the rest that we have to talk about. Look forward to it. Take care. Thank you.